Pleased to be joined today by Dr. Scott Grafton. He's a distinguished professor of psychological and brain sciences at the University of California, Santa Barbara, where he directs the Action Lab, studying the neural basis of goal-directed movement. Scott, welcome to the Nature and Nurture podcast.
Thank you for having me. I look forward to this.
Scott, what got you interested in the neuroscience of movement?
I think, first off, I'm a neurologist. I'm a very physical person. So you put those two things together.
Physical, like exercise and outdoor activities?
Yeah. I'm trying to not use the word exercise in my vocabulary because it's almost a pejorative for most people. I'm a strong advocate of physical activity as a way to think about what we do in the natural world. And and in neurology, a good portion of what you see in terms of patient care is people with movement problems, either movement disorders or stroke or what have you. And so that becomes a prime problem. And when I started all this, with imaging years ago, we had a very naive notion of sort of how we go from thoughts to actual movement. We had a lot of people studying muscles and spinal cords and nerves. We had a lot of people studying decision-making and cognition, but like, how do you put those things together? How do you go from idea to action? And so that plus the patient work really motivated me to sort of develop this career.
How much of that can we learn uniquely from humans, as opposed to, like, if you want to understand the fundamental mechanisms of movement, you need to do it at a very basic, maybe cellular level, starting with simple organisms and moving up to humans?
Yeah, it's funny, a good example would be like sequential action, right? So doing any kind of sequencing of behavior. We studied classic sequential learning problems in humans for decades, and then a colleague came along at UCSB and she studies sequencing in the fruit fly, right? So fruit flies have these beautiful sequential actions, sort of grooming behaviors and things like this. They do itching and grooming behaviors. And they also have these little dance kind of movements they do as well. So they're super simple organisms, right? You can get at fundamentals of sequencing. I mean, clearly there's a gap between the two. You can really, with the fly, you can really get down to like specific neuronal chains that would generate a sequence. Can't do that in people. What you can do in people, though, is look at the flexibility of sequencing and generalization, sort of more broad types of learning mechanisms. So they complement each other, but they certainly don't necessarily overlap.
What do you mean by sequencing?
Typing would be a good one, or playing a piano arpeggio, right? Just with your fingers. That's been some variation of that has been sort of the hallmark of sort of human research for decades. The serial reaction time task is a good example of it, where you learn to type out sequential movements with your fingers. In some ways, we give too much credit to that kind of sequencing, in our human studies. It's just so easy to have people do things with their fingers, but we forget we're sequencing all day long. We put our underpants on before we put our pants on, and then you put your belt on, right? There's so many sequences in everyday life that are sort of baked into our behavior through reinforcement learning.
Can you tell in the brain the difference between someone sort of acting out this unconscious sequence, like whether it's the underwear before the pants, or if you're an expert pianist going through the motions, as opposed to that effortful type of motion, even if it's the exact same movement?
So if you think about the evolution of imaging, So what we were doing, let's say in 1988, it was just, could you even see a change in the brain over time as a person learned a simple action, simple skill, like pursuit rotor, learning to move their hand in a circle, or learning to type something out consistently. Would you even see anything change in the brain? And certainly you do. The interpretation of what those changes actually mean is taken a long time sort of to unpack. And then you get into the whole issue of stages of learning. And this goes all the way back to Harker and Bryant from 1890s when they studied people learning telegraphy, like Morse code and telegraphs, right? And you could see people go through stages. So they'd have a sort of a logarithmic learning curve, and then they'd plateau, and then they'd have a second curve and a third curve, and they'd bounce their way up. And they introduced this whole idea of stages of learning. And so you put that on top as well, and you realize even the simplest sort of sequencing tasks have multiple sort of types of acquisition that are occurring in parallel on different time scales. And that's taken a long time for people to sort of get their head around, but I think it's really important. The idea being like, okay, you want to type a phone, you want a new phone number, right? Someone you really, really like, and you memorize it, right? So you're immediately going to use working memory and explicit memory buffers to fast track that. Then you also have this novel phone, so that let's say you have a new iPhone, and so you got to figure out all the interfaces of your body with a device. So you've got all that learning to do. And then now let's say you've typed the number in 10,000 times, right? It's a whole different, we have automaticity, right? You're no longer even using working memory or engaging in the problem of interacting with the device. Your hands are just executing, right? So they're in a simple case, you've got three completely different kinds of learning going on. We used to say it occurs in sequence, but I don't think it's sequential. I think you throw everything at the problem in parallel, right? You're using working memory, you're trying to automate it, you're trying to get, familiar with this device. All these are different kinds of learning that take place in parallel. And
Is a reflex
they just operate on different time scales, right? Some are fast and some are slow.
Is a reflex fast response, like moving your hand away from the stove, or the knee jerk you get if you hit the right spot on the knee, is that an example of something, a movement that's learned and just learned very early in development? Or is there such a thing as instinctual behavior that can bypass learning somehow?
Well, reflexes to pain are pretty hardwired. A baby will withdraw to pain, right? So that's pretty much a spinal cord reflex that's baked in development. What's interesting is sort of the second wave of learning that occurs in children, and Karen Adolphs is really the pioneer in thinking about this. Basically, babies as optimal experimenters, right? So there's far less innate knowledge of how your body fits into the environment than we take for granted. She shows that like a crawling infant will just try crawling down a steep incline that's way too steep for it to make it down safely, but it has to figure that out through experience. So she sets up these crazy slides in the lab and lets the kids essentially have fall down, but of course they have parents and everybody there to RAs to catch the kids and so forth. But it just shows that like this idea of they have this innate fear of edges and falling and this sort of thing. That's not really the case. They're little experimenters, and they're trying to figure out how their bodies work, in the environment. So it really goes back to sort of the Gibsonian idea of affordance and like, how does my body interact with with
Your
everything around
research,
me.
is it all in adults?
Mine is, it's all in adults. Yeah.
Mhm. So how can you disentangle what is like a learned behavior over the course of your entire lifespan leading up to the time that you're testing them in adulthood versus anything that's new that you're hoping to teach them in the lab?
Well, this is, yeah. So this is a killer problem. We faced quite a while ago. And you know, like one way to think about it is, all right, we were really interested in how people interact with objects. Okay, reaching and grasping and interacting with objects. Now, the problem with an adult is, you know, we've already handled tens of thousands of objects, you know, probably millions of times. So our experience level is through the roof by the time we're an adult. And so what does it mean to try to look at novelty in that space or learning in that space? It doesn't come easy. You can cook up really weird objects, but you know, we kind of put that to the side and said, look, if we really want to understand, because what we were really interested in was, as you acquire skill, does it change your perception of that action, right? So if I've learned to do something novel, will that change how I see the world because I now have physical experience? And the motivation for this actually came from the Olympics. I was at the Atlanta Olympics. What the only tickets we could get were at the 10-meter high diving. Okay, you know, the really high platform diving. So you're up there and sort of in the wind, even with the divers. I don't know anything about diving, right? So I'm watching them dive, and it's like all of us when we watch on TV, you know, you just look at the splash. You know, if it's a small splash, you think, well, it must have been a good dive, right? You really don't know much because I'm not a diver, right? I've never done this. Everybody in the audience around me was a diver, and they were seeing things, right, and describing things constantly that I'm like, really? What just happened? What did you see? So it's clear that their perception of that physical action was different than mine through their experience. So how do you nail that in the lab? We thought, well, we can't do that with hand object or typing because we've done too much of that with our arms. So we have to go somewhere novel. And so we switched to dancing, and we did a whole series of studies in the around 2000 in dancing because that's really interesting because you can have, you know, you can learn a modern dance piece that's absolutely specific. It's really hard. It takes 6 weeks to learn. You can perform it at the end in front of a large audience. You can watch other people do that dance. And then you can also have parallel dances that you're not learning. And so we did this. A group in London came up with the same idea at the same time. So they had ballet dancers watching capoeira dancers and capoeira dancers watching ballet dancers. We kind of started from scratch and had modern dancers build up a new repertoire, and showed that, yeah, as you learn physical skills, your perception of the world of those kinds of actions does change. And then we replicated it with knot tying. So as you learn novel knots, as you see knots, all these physical or motor systems are engaged, just seeing those familiar knots versus unfamiliar knots.
I remember an old debate in this idea of how perception is filtered. I don't remember what it's called, but on one camp, you had the idea that your brain is soaking in all information and then only afterwards filtering out what's unnecessary. And there's some sort of top-down mechanism there. And then in the other camp, it's like if something seems irrelevant, it never even gets perceived in the first place. So like in the dancing example, maybe you're noticing that someone's ankle is at like a different angle than it should be or something. And it's like in one version, you might say everyone notices that, but then just quickly discards it as irrelevant. And then but you might also say you're never even going to look at the ankle specifically unless you think you know that there's something worth attending to there.
Yeah. Yeah, I mean, the danger of what you just proposed is it's your classic straw man. It has to be attention
Uh-huh.
works one way or the other. And there's one thing we've learned about attention is, well, it's a lot of things, and it's really flexible. And you can have both of those things occurring, right? So we can be dealing with salience, pop out effects, top down control, bias. We can be filtering both early and late stages. I mean, all these things going on at, simultaneously with attention. So it's a little hazardous to say, well, we do it one way or the other. I think with certain, I mean, just going way back now with thinking about motor systems, it's sort of in this space. I mean, when I started, there were basically people studied vision, people studied attention, and then people studied movement, like how the motor cortex makes muscles twitch, right? And those communities didn't talk to each other at all. They have different study sections at NIH. They have different meetings, right? And one of the very first things sort of we discovered with imaging and in parallel was found in single unit recording in monkeys was a lot of the classic premotor areas respond to visual stimuli. So, this hard line of vision in the back and motor in the front, right? It doesn't work, right? It's just not true. You have a lot of systems in the brain that are multiplexed. And so, that led to this idea of mirror neurons as one explanation for why you would have sort of motor areas responding to the perception of action. There are other explanations. You don't have to invoke mirror neurons, but I think the more important thing was it completely broke down this sort of bifurcation of trifurcation of the brain into these different systems. It's much more multiplexed than we realized, and so in the motor space, what you see is if there's a graspable object in front of you right now, I mean, if you just look at any object in front of you on your desk, your premotor areas are already planning ways they could grab it, ways they could interact with those objects. So they're way ahead of your, like, shall I pick up this pencil or not? They're already evaluating the pencil and what the potential affordances are for interacting with it. So there's a lot of feed forward, adaptive processing going on. So that plays into attention, because now you can say, well, part of what attention does is it says, well, the pencil might be relevant, but I'm not going to pick up the computer screen. It's a little too heavy and pain in the neck.
And there are separate brain areas for describing versus actually doing something, right? Like if you have one type of brain lesion, you can describe the function of a lock and key, but you might not know how to actually manipulate them. And then if you have a different type of lesion, you'd be able to do it, but not describe what they are.
Yeah, so this goes back to Lietmann all the way in 1911 his patient was named Mr. T, which is kind of fun. Mr. T was a Viennese administrator. He had a stroke and he had an aphasia, so he couldn't talk very well. And he had paresis of his one hand, but he could comprehend. So you could say, Mr. T, go to the window and look out. He would get up, walk over and look out the window. So he's clearly understanding, you'd say to him. Now you could say, okay, with your good hand, show me how to use a hammer. And he would, he would have some strange movement with his hand, or show me how to use a comb. And he couldn't demonstrate how to do actions. And that was generally associated with a left supramarginal gyrus parietal lesion. And there's been tons of studies since showing that's sort of the sweet spot in the brain for this kind of apraxia. We've had a lot of imaging studies showing this over the years. So that's sort of the first cut. And then you get into slicing and dicing sort of the syndrome into, well, is it conceptual? is this problem a conceptual one? So some patients, it seems to be more conceptual. You know, it's like, what is a hammer? Or if you show them a bunch of tools and say, which of these would you use to solve a problem, they wouldn't know which tool to use. So they're sort of at high level. So you get into semantic kinds of knowledge all the way down through, yeah, they know which tool to use, but their hand just doesn't sequence, you know, the movements right with the object. There's some that are quite paradoxical. They can't show you how to use an object, but you put the object in their hand and they can use it perfectly. So there's all these different subtypes, which tells you, yeah, there's modularity within this system, spanning all the way from very high level semantic, cognitive, all the way down through fine-grained sort of sequential specific movements you'd have with the
What you
executing an
see
action.
with those early lesion studies is replicated in later neuroimaging studies. The same areas have the same functions.
Oh, totally. Yeah, that's been one of the sort of the nicer sort of narratives over the last decade or two, is really strong consistency. And it's still going on. We've got some recent papers, you know, like one of the issues that comes up is, so if you think about where semantic knowledge really sits, like people with semantic dementias, they tend to be where they lose knowledge of what things are. It tends to be left temporal lobe. And left temporal lobe is also really important for recognizing objects. Now, the supramarginal gyrus in the parietal lobe is this area for apraxia, right? For sort of going from idea to action with your body. So it turns out there's this really strong white matter tract that connects inferior temporal cortex up with SMG and related regions. And so now you can, with sort of state-of-the-art connectivity imaging, you can sort of find patients where there's a very specific disconnection between these two parts of the brain. So your ability to sort of look at an object, understand its texture, what its shape is, what it's for, what its meaning is disconnected from how you would use it.
How do you know how to interpret the different strengths of your findings? So, for example, more connection or more brain activity could mean more function, but you could also tell the opposite story. You could say something like less activity that shows up on the scanner means greater efficiency. So that's actually what you want.
Oh, that's a great question. Yeah. So here's an example. Here's a really simple example. One of the very first diffusion scans that was done, we did it at Dartmouth, and we looked at corpus callosum fractional anisotropy, and the degree of connectivity between essentially the two hemispheres in a reaction time task, intermanual reaction time task. And so you could say, is more connectivity going to make interhemispheric communication faster or slower and lead to faster or slower reaction times when you're trying to push information between the hemispheres, right? And you don't know, right? It could go either way. It's a complete bet. And it was counterintuitive. The results were counterintuitive, but it is what it is. And so that keeps coming up over and over. It's like, is more anisotropy or more white matter integrity, is the jargon people like to use, going to lead to faster, slower cognition, motor behavior, visual processing, what have you. And it's all over the map. Part of the reason it's all over the map is really specific technical problems with the measurement itself, but the other is just conceptual. I think the way around the connectivity work, what we've been doing is it's much simpler. You basically take the human Connectome project data set, which is, you have like 1,000 brains, okay? So you have a really normative data set. And now I can take a patient who just has like a regular MRI that was done in the hospital. And so I know where the lesion is, and I know what their deficit is, and I can project that into the Human Connectome project and say, what's the most likely disconnection given my 1,000 people? I have a pretty good probabilistic estimate of what brain areas are disconnected from each other and the severity of those disconnections. And so you can relate their their simple lesion with a good estimate of what areas aren't talking to each other. So that kind of inference is pretty clean. The harder one, I think, is when you get into the functional space. This goes, we dealt with this with Parkinson's years and years ago. So if you have a Parkinson's patient and you have them move a joystick at a standard rate, right? And you compare that to normal people moving it at the same velocity, you get many more brain areas activated in the Parkinson's patient, except for what you would normally see. Like the motor cortex is underactivated, the supplementary motor area is underactivated, but a whole bunch of other areas are overactivated. So what is that? Is it compensation? Is it accommodation? Is it, is the brain reorganizing? What's going on, right? It can be really hard just in that simple case to know what's going on in the patient. Now, what's interesting about them is if you give them dopa, L-dopa, or they've got a stimulator and you turn the stimulator on, things all normalize they converge towards the normal pattern. So that's a case where you can sort of make an inference, that says, well, probably all this overactivity is a compensatory strategy of other brain areas just trying to boost performance, sustain performance when the prime areas are underperforming. But that's not, so it works in that case, but not necessarily, that's not necessarily going to be the interpretation for every neurodegeneration. It came up early in Alzheimer's as well. Some of the very earliest Alzheimer's metabolic studies, where you've got hippocampal degeneration, loss of cells in the hippocampus and temporal lobe from Alzheimer's disease. They actually show overactivity of glucose metabolism in their residual hippocampus. Same idea, as if whatever's left is working overtime to try to sustain performance. Right? So it can be very hard to make strong connections between over or under activity and what the brain's actually trying to accomplish. As a clinician, you sort of get the impression that brains have this incredible ability to try to throw every resource they can at a problem to functionally accommodate whatever deficit you have. They will use every trick in the book to sustain performance. You see this in stroke patients. They got a paretic hand. Well, they'll learn to hold a jar with their upper arm, and they'll squeeze things in weird ways, and just their body will naturally find unusual solutions to sustain performance. So in the motor domain, we see functional adaptation all the time, and I think we do the same across all sort of cognitive domains. It's just harder to map.
I know in young children, there's a lot more neuroplasticity and it's easier to build those compensatory mechanisms. But do you see the same thing with age in adulthood? Like, would it make a difference if you have a stroke at 30 or 40 or 50?
In general, the older you are with a stroke of the same size, let's say, that same magnitude, the worse you're going to do. That's pretty much universal. But once you hit the age of 7 or so, 6 or 7, you're going to be limited regardless. you're going to be hit pretty hard with a significant size stroke.
Is that an earlier age of 7 because motor area or perception areas of the brain are much more primitive and mature earlier? Because you hear a lot about your brain is still maturing well into the 20s, but that's more like frontal cortex
frontal
regions.
cortex, yeah. So a great example would be a child with or an infant with what's called infantile spasms. So they have really severe seizures that you cannot control with medications, and they might have it driven by something called tuberous sclerosis, which is basically malformations. So you have these nodules in the cortex that are just causing seizures all the time. And the kid won't develop cognitively because they're essentially either having a seizure or recovering from a seizure. So they're out till lunch. And if they're lucky, all the abnormalities are in one hemisphere. And so you can take that hemisphere out. Now you leave in the basal ganglia and the thalamus. You're just doing a cortical resection. And it's quite remarkable. So you do that in a 2-year-old or a 3-year-old, really up to a 4-year-old. 20 years later, their speech is going to be normal, affect, everything about them is going to be normal, except you'll find that their contralateral hand will be a little bit spastic. So the fine hand dexterity is lost. So you really do need your motor cortex for dexterous movement of the hand. And their walk might be a little bit stiff. So that leg, the contralateral leg, will have a little bit of a stiffness to it. But other than that, they look really good. And so it fits with your idea, right? That the motor cortex comes in early, and you're trying to, it's committed to learning dexterous movement, as soon as you're born. So that system is locking in pretty quick, but everything else is pretty flexible. Actually, like 6 months ago, I had we do a lot of scans and undergrads, and I had a volunteer come in for study, totally normal, honor student, great person, come in, put them in the scanner. They don't have a left temporal cortex. They don't have a left temporal lobe. It's just not there. It's missing.
Wow.
Come on out. We'll show you your scan. We got to talk. And and they have a congenital cyst. So just when the brain formed, this cyst formed. The left temporal lobe never developed.
So this wasn't a surgery that they intervened with early on. This is just, it wasn't there.
No, it just was never there. Yeah, never. Yeah. And so they're a beautiful example of something. And because it didn't reach up into the motor areas, they didn't have any motor deficit either. Now, I bet if like the neuropsychologists will tell you, well, if we really test them, like really fast semantic tests, or, really sophisticated language testing, we might find some deficits, but in general, they're subtle, right? In terms of that person's ability to function in the world, you're not going to notice anything.
So if you don't notice any functional deficits, what comes out of we need to talk? What do you actually do? If you find out that you think you have a completely normal brain, and then it turns out a piece is missing, but you're doing fine.
Yeah, this is institutional specific. Each research center that's doing fMRI research will handle this incidental findings differently. We're not a medical center. We're not doing clinical diagnoses, but we do put in our consent forms. Look, if we find something significant, we'll inform you, right? If there's something obvious there, we'll let you know. Now, there's a fine line. Like, there's a lot of people with, like, the base of the skull might be a little bit enlarged, and there's a little more spinal fluid there, so it kind of stands out. You see that all the time. So it's like, so what, right? We don't even. It's just like it's like having a big thumb, right? So sort of there's a limit to how severe things need to be. Now, when someone's missing a lobe of their brain, To me, it's important to tell them, because like, if suppose they get in a car accident or something and they get a CAT scan and they don't know about this, and everybody's like, oh my God, look at this. They already know that that's there. So it'll, for their future medical care, it really allows them to sort of do things wisely.
I see.
Yeah.
And this,
And for students, it's fascinating, right? I mean,
yeah,
it's like,
if you're
they get to
missing
learn.
a piece of your brain, is there literally a hollow spot, or does it fill up with spinal fluid or something
It's
else?
always spinal fluid. Almost always. It's like it's like a little membrane filled with spinal fluid that connects in with the ventricles in some way. Yeah.
Does that make you more susceptible to concussions or things like that because there's this fluid region that can be shaken around more easily, or there's more room for other regions to get shaken into?
That's a great question. And I don't know, because it doesn't happen enough to know, like, we just don't have the evidence to know.
Mhm.
Um, but it is a strange problem. And like, when in those 1-year-olds or 2-year-olds, when they do the hemispherectomies, you have this problem of, right, I've just taken out half your brain. So what do we put in there to fill the space? Because in that case, you don't want, things really could jostle around, right? You don't want, so there's, like, you put in ping pong balls, hydrogel. There's a whole surgical literature on what you would sort of fill in that space. But generally water is a fluid filled bag of water is a pretty good cushion.
Mhm.
So for like risk of concussions and things like that, I think they're in pretty good shape.
You mentioned working on the Human Connectome project, HCP. Do you happen to work with Randy Buckner on that?
I have not, no. I've just benefited from all of his hard work
Uh-huh.
putting a lot of
So
that together.
I
Yeah.
know that Randy's has a large role in HCP, but he's also now moving towards these individual focused studies. So, like, rather than doing one or two scans on a whole bunch of people, you're doing a whole bunch of scans on very few people. And I guess there are trade-offs there because when you average across many individuals, there's noise in the data and the whole brain picture gets fuzzier. And you can compensate for that by recruiting like thousands of individuals, like what you're doing in HCP. But then the polar opposite way of compensating for that would be like just focus highly specifically on individuals with many scans. So could we talk about the pros and cons to each of those, and which one comes up more often in your line of research?
Yeah, I feel like I'm one of the leaders in that strategy of doing dense sampling. Um, Certainly Russ Poldrack was one of the first to do it. I'm going to scan myself. I don't know how many times he did it. Years before that, we had done a learning study where we had this really hard mechanical task where you had to, while you're in the scanner without any vision, you had to put a bunch of nuts and bolts together on this board. So you had to, we called it the Vermont mechanics task. It's like you're under a car trying to put a bunch of nuts and bolts together, right? Really hard. 5 of us scanned ourselves doing this for a year. Right. And this probably, we did that in 2000. Right. So this is long before everybody else was thinking about dense sampling. So my original motivation was for learning studies. If you really want to look at long-term sort of development of automaticity, let's do it over this really, really long time horizon. And then we had people come back 6 months later, right, to look at retention and so forth. We never published it. It was an early crappy scanner, and it was kind of noisy data. I was never satisfied with basically the quality of the data. So we never published it, but it led me to think, yeah, let's just do some dense sampling, especially in learning, where you're really trying to, within an individual, what's really changing. And then a couple of years ago, Lauren Pritchard, she was a new graduate student at UCSB. She's really interested in how the menstrual cycle impacts brain activity in healthy women. And so we said, well, why don't you just scan yourself every day for a cycle. Like just, okay. So we did it, right? And then again on oral contraceptives. And I can give her name because she wrote the papers. We all know it was her. That's the subject. And yeah, so it's really neat because now people had sort of sporadically looked at sort of the effect of estradiol and progesterone on sort of cognitive systems. But here you've got every single day, right? So you can really look at network dynamics and structural changes in exquisite detail. And those are really successful studies. And so they call that the 28andMe project. And they're still doing it with, they're doing it in men. They've got a whole bunch of variations to it. So I think the question now is not whether it's like better or worse. It's like it's a ton of work, and so how do you maximize the actual effort? And I think it's important to really drill in on specific questions. Like Russ's work was, I think he did a variety of tasks, and he was really interested in just natural variation. Lauren's was sort of different. It was like exactly how does estradiol impact hippocampal size, or progesterone impact hippocampal size, or network dynamics and resting state activity. So I think you want to go in with specific questions. I think that sort of the natural variation question you can do, but it's not nearly as interesting as having a specific perturbation you're tracking in parallel.
Do we think a lot about brain function changing day to day, or hour to hour, minute to minute, and maybe even connectivity? But it's interesting to see such short-term structural changes.
Yeah, it's really surprising. We have a study right now. We've got 30 women. It wasn't a dense sampling every day, but we have them scanned at peak ovulation, luteal, and then during their periods, luteal phase and menstrual phase. So it's 3 hormonal extremes, and there's some wild changes in white matter, hippocampal density, their size, resting state activity, all you name it. Whopping effect. I mean, we all kind of knew this. Women will say, depending on where I am in the cycle, the way I think changes. And these are just, some structural and functional correlates of what people have experienced.
Do we have
centuries.
any idea of the why yet?
Yeah. So some of it's, there's all these sort of you can take the evolutionary standpoint, right? You want the estrogen to make you think, or during peak ovulation, you want to be thinking in terms of mate selection or not, right? There are all these sort of just those stories you can put on it.
Yeah, there's studies on attractiveness where it's like during ovulation, I think women are more attracted to like certain masculine features, and then off ovulation, the opposite.
Yeah, and then, but yeah, so that's one sort of extreme. The other is just you have to remember these hormones have widespread effects on just about every cell in the body, and they're not just for cognition. So here's an example. We see dramatic changes in white matter anisotropy using really state-of-the-art gradient tensor imaging techniques. Basically increased anisotropy, increased volume. So there's essentially more water that's in a constrained compartment in your white matter when estradiol is high. Why? It probably has no functional relevance to mate selection or anything like that. It's just probably estradiol influences aquaporin 4. We know that's true in animal models. There's just different amounts of water in the brain. That water has no place to go. The anisotropy goes up. So you got this really simple, non-psychological explanation that's just
Right.
the effect of just the hormone in the brain. So that's the that I like that contrast, because it not everything has a psychological explanation, right? I mean, or it needs to be have a evolutionary psychological explanation. It can just be complicated hormonal interactions with local receptors.
Absolutely. Did you see a dense sampling study from a couple years ago? They were scanning people's brains every day, and then they casted their arms for a couple weeks, and they took off the cast, and they were looking at motor changes day to day.
Yeah. God, I can't even remember the results. The flip side of that was what motivated that was the forced use model in rehabilitation therapy. You've got this problem. So if you have a stroke, one of the things, let's say your motor cortex in your right hemisphere, so your left hand's not working very well. One of the problems in recovery is that the good hemisphere drives inhibitory inputs into whatever's residual in that damaged hemisphere. So you have excess inhibition projecting onto this system that's trying to recover. Okay. And that's aggravated by using your good hand all the time. Right.
Mm-hmm.
And so the idea is, if you've got a weak right arm from a stroke, put the left hand in a cast and force use of the impaired hand. And in doing so, you change the amount of inhibitory input that goes into that side whenever you're using the good hand.
Mm-hmm.
So that's what led to those kinds of studies. It was like, well, will we see these changes of interhemispheric inhibition when we in a healthy person, we just stick a hand in a cast?
Yeah, I remember the functional connectivity between left and right motor regions went down during the casted period and then rebounded after they took it off. And there was another thing, they called it spontaneous disuse pulses, as in whatever arm region was casted, they saw just like random bursts of activity, and they were speculating whether those were meaningful or whether it was an attempt to like rewire and form some sort of new function since they couldn't be used anymore, or something else.
Yeah, you have to be careful. It's not just your arm being twitchy in the cast, right?
I didn't think of that. Yeah, that'd be
Right.
interesting.
Because yeah. But yeah, these are really interesting ideas.
Mm-hmm. I want to close, Scott, asking you what you're most excited about, either in your own research or science as a whole.
So what a colleague, Peter Strick, and I are really zooming in on the question of how do you really characterize allostatic mechanisms in the brain? And this gets kind of into your nature-nurture theme. Yeah. So we all learn homeostasis and physiology, right? Which is basically the thermostat on most of the physiology of your organs are explained in terms of like a thermostat on the wall. If glucose is too high, you make it lower with insulin, and blood pressure is regulated down if it's too high, and so forth. So we've got these feedback mechanisms to regulate all our organs. And that's homeostasis. And then Peter Sterling in the 90s coined this term allostasis. And the simplest way to phrase it is have anticipatory changes just enough, just in time. So the simplest example is stand up, right? And you don't want to stand up, have your blood pressure drop, and then react to that. You'll pass out every time you stand up. So before you're even standing, you're anticipating the physiologic requirements of the standing movement. Um, a horse, actually, that's not a good example, but giving a public speech, right? That's another good one, right? Like an hour before you give the speech in public, if you're not used to doing this, your heart's already racing, right? You're anticipating either a threat or a challenge through your sympathetic responses way, way ahead of time. So now the game is understanding how cognition in the cortex is reaching all the way down into all your organs. Like every textbook just kind of shows your brainstem and all the reflexes going up to your brainstem. And it's like, yeah, the brain might be doing something too with your physiology, but whatever. We don't care about that. But that really matters, right? And it's a real description of health. So if you think about the social determinants of health, stress, like chronic stress, why does that lead to hypertension? Why does that lead to, lots and lots of health problems? Why does it lead to adverse non-adaptive behaviors? A lot of those are really just disrupting out normal allostatic mechanisms. And we know that physical activity is one of the most powerful ways of sort of resetting allostatic health.
What does that mean?
Allostatic health or physical activity?
Or the whole process, like how does it reset this allostatic health?
Well, we don't know, but it's certainly the case that a 20-minute walk in the woods is probably more effective in controlling blood pressure and stress than almost any medication I give you. And so there has to be a relationship between your cognitive appraisal of that environment and the physical engagement you have in that environment and what all your organs are doing to adapt to that environment.
Very interesting.
Yeah.
Thank you very much for your time, Scott.
You're welcome. Hope this was useful.